Next question, GATE 2022 Life Sciences, Section S Microbiology, question number 81. Which of the following statements are true for archaea? A. Cell wall in archaea contains muramic acid and D-amino acid. B. N-formyl methionine is the first amino acid to initiate new polypeptide chain synthesis in archaea. C. Methionine is the first amino acid used during protein synthesis in archaea. And D. Membrane of archaea contains phytanyl rather than fatty acids. So we all know that we have prokaryotes, eukaryotes, and then we have archaea, right? These are the main three divisions: prokaryotes, eukaryotes, and archaea. So archaea are not classified into prokaryotes because they have some different, these are the more primitive type of organisms. They live in very harsh conditions, hence they were considered as most primitive type of organisms as compared to that of prokaryotes and eukaryotes. So they are not completely as same as that of prokaryotes. They have different composition in their cell wall. The cell wall composition is pretty much different. And also the environmental condition in which they are very harsh. So and they are primitive to that of bacteria. They are very primitive to that of prokaryotes. So their cell wall is very unique and different. So let's first go into that. So this is our bacterial cell wall. We have gram positive bacterial cell wall and gram negative, like we discussed in the last lecture, right? So in gram positive we have peptidoglycan layer, we have teichoic acid, right? And in gram negative we have lipopolysaccharide outer layer and very thin layer of peptidoglycan, right? Because of this differentiation we are able to easily do gram staining and identify gram positive and gram negative bacteria, right? That is what we studied in the last lecture. So this is the overall structure of bacterial cell wall. Then we have archaeal cell wall. So archaeal cell wall, so they are not broadly divided into, they don't have a specific composition of cell wall. So this is very important, that the archaeal cell wall composition mainly depends upon the different species of archaea. Different species of archaea has different composition. So as you can see here, we have methanobacterium and here we have methanococcus halobacterium. The methanococcus and halobacterium, so they are both cell wall composition is very different. So we have a phospholipid, bilayer, and they have heteropolysaccharide. This is very important. They have a heteropolysaccharide and they have a pseudo peptidoglycan, which is also known as pseudomurine. So they have murine, pseudomurine, also known as so this is found in mainly gram positive. In gram positive also we have murine. So they have pseudo peptidoglycan, which is comprised of murine like structures, and they also have heteropolysaccharide. Remember polysaccharides are sugars, and what are heteropolysaccharide means? Different. We have hetero and we have homo. Homo means there is only one single subunit, and in hetero we have multiple subunits, right? We have multiple subunits of saccharides. So and in methanococcus and halobacterium we have so the phospholipid composition is also different, and there is a S layer. So this S layer consists of various different just like pseudomurine, it has this S layer is composed of other other glycans, just other sugars. Okay, and then this is very important so fatty acids in prokaryotes, and eukaryotes, right? Okay, in humans also, humans, plants. So we have fatty acids like this. Okay, so we have a ester linkage. Okay, so this is a ester linkage, CO bond O, right? This is the ester linkage, and then we have long chains of acyl chain. So the fatty acid acyl chain is ester linked in prokaryotes and eukaryotes. Okay, but in that of archaea it is ether linked. Okay, in archaea we have ether linkages. This is very important distinction. I'll show you in the next slide. So in bacteria and eukaryote, as I mentioned, so this is the glycerol backbone, right? This is a glycerol backbone, and to this glycerol backbone we have this OH group is replaced by acyl linkage. So this is a ester linkage. C double bond O is a ester linkage, right? C double bond O is a ester linkage. But in archaea we have a ether linked. Okay, we have ether linkages. So this is the ether linkage, CO bond C. This is the ether linkage, and this is the ester linkage. Okay, this is very important. So we have ether linkages in archaea and we have ester linkages. So all the fatty acids in bacteria and eukaryotes have ester linkages. And another important modification is phytenyl side chains. Okay, it has more alkyl groups. Okay, branched alkyl groups. So this branching of acyl chains is seen in the archaea. So why do they have this modification? As I mentioned, archaea are found in the harsh conditions, right? They are like Methanococcus or Halobacterium. They are found in harsh condition with various high salt concentration or high temperatures. So these phospholipids, branch phospholipids and ether linkages. So ether linkage is pretty strong as compared to that of ester linkage. Okay, so ester linkages are very weak. So ether linkages are pretty strong and sturdy. So in order to protect the bacteria, the membrane, the phospholipid bilayer membrane is of ether linkage, and also we have phytenyl side chains. Okay, we have branching. Okay, we have branching in the fatty acyl chains. Okay, these are the important classic difference in the phospholipid of bacteria and eukaryote and in archaea. And this is how the phospholipids, this is how they form. So so we have a bilayer, and we have distinct space between the two bilayers, right? But in archaea we have a connecting chains between the head groups. Okay, we have connecting chains of this phytenyl side chain has the connecting linkages between the head groups. These are the head groups, right? So these are the distinct features of archaea and phospholipids in case of phospholipid bilayer in archaea and bacteria and eukaryote. Okay, so coming back to the question, cell wall of archaea contains muramic acid and D amino acids. So D amino acids are not present in archaea. Okay, and we don't have muramic acid, but we have pseudomurine, right? Pseudomurine is not same as muramic acid. So muramic acid and D amino acid. So in bacterial plasma membrane we have in peptidoglycan. So peptidoglycan is nothing but sugar molecules, and then we have D amino acids attached, right? We have alanine, D alanines, four amino acids are attached, and this is how the gram positive peptidoglycans are made, right? So D amino acids are not present and muramic acid is also not present. We have a pseudomurine, okay, in archaea. The next difference is N-formyl methionine is the first amino acid to initiate new polypeptide synthesis in archaea. So this is also one of the important difference, that in bacteria we have N-formyl methionine, right? F-met amino acid as the first amino acid, and in eukaryotes we have only methionine, right? We have only methionine. So in prokaryotes we have formyl methionine, and in eukaryotes we have methionine. In archaea we have only methionine, okay? We don't have formyl methionine, okay? So this is wrong. This is also wrong. There are no D amino acid present in the archaea, and also we have pseudomurine. So methionine is the first amino acid during protein synthesis archaea. Yes, this is correct. And then membrane of archaea contains phytanyl rather than fatty acid. This is also correct, right? We have phytanyl side chains. So the correct options are C and D.